Throughout nature, symmetry is present in many forms. In bilateria, the characteristic body plan consisting of one central axis of symmetry is often broken, as is the case in the signature swirl of the snail. The snail is a prime example of a chiral animal, that is to say an animal which is not a mirror image of itself. There are advantages and disadvantages to both symmetry and asymmetry, and depending on the circumstances, natural selection will favor one over the other. In outward appearance, symmetry tends to be associated with high genetic fitness and is often preferred in many examples of mate selection. However, internal mechanisms may benefit from asymmetric development. Specializing certain regions for different tasks allows for a more efficient resource use and improved fitness overall.
There is a study from 2000 in which two groups of chicks, lateralized, meaning that there was an asymmetry to their brain that allowed it to be specialized for particular functions, and non-lateralized, whose brains did not show this asymmetrical specialization, were exposed to a stimulus while feeding. The brain development of the chicks was controlled during embryonic development, where the chicks who were exposed to light for a certain time interval would then develop lateralized brains, whereas the chicks incubated in the dark were non-lateralized. The stimulus used during these trials resembled a raptor, and experimenters measured the time that it took for these two groups of chicks to detect this quote-unquote predator. Results showed that the lateralized chicks show a significantly shorter delay in detecting the stimulus versus the non-lateralized chicks. There was also an advantage to left-side stimulus for the lateralized subjects, suggesting that the right hemisphere of those chicks was responsible for this specialization. The results of this study support the advantages that asymmetry in the brain can provide, and that there is increased efficiency in performing the dual tasks of feeding and vigilance to predators at the same time, which gives a clear evolutionary advantage to those organisms displaying this type of asymmetry.
The scientific paper on left-right asymmetry describes the impact of the gene formin on body symmetry, in which the gene creates the dextrodevelopmental structure in snail shells. This asymmetry is a critical, if not ancestral, component of animal development. Not only can this be seen in the chirality of a snail shell and cell cleavage in frogs, but asymmetrical development is common throughout much of the biological world. Fractal development, a geometric patterning that results in a repeated segment of itself, is often described as having expanding or evolving symmetry. Despite the inherent symmetry of the pattern itself, it often results in asymmetrical branching of growing parts. This patterning is responsible for many natural phenomena and can be used to describe anything from the veining in a leaf to the budding of a Romanesco, a kind of broccoli. What does this mean for the symmetry of living things, however? In a paper by Rob W. Glennie on the emergence of matched airway and vascular trees from fractal rules, the symmetry and asymmetries that result from the fractal development of the fetal lung are discussed. The original development of the lung has been well recorded as an asymmetrical budding of 2 lobes on the left and 3 on the right. This was traditionally thought to develop in symmetrical pattern along 3 different axis planes of branching domain: branching, planar bifurcation, or orthogonal bifurcation. However, this confers several advantages to fractal development that results in asymmetries. First, like formin, small proteins that are genetically selectable and inheritable can be used to develop this patterning. Secondly, these asymmetries maximize the available 3-dimensional space of the lungs, and small asymmetries within successive branching of these pulmonary trees create broad distribution of blood flow and ventilation at the terminal segments. An important disadvantage is that the small asymmetries that are repeated at each branch bifurcation create high levels of variability in blood flow at the terminal branches, which is only expanded upon by the next branching, leading to severely impaired gas exchange. Therefore, having structural extracellular complexes that direct symmetrical development can be very important. Despite the benefits that asymmetries can bring, extensive asymmetry can lead to deleterious effects.
A 1992 study of Swedish barn swallows examined the ways in which asymmetry affects female selection of a mate. To do this, the authors of the study colored the ornamental tail feathers of male swallows with correctional fluid so that the feathers of some birds appeared asymmetrical. They found that it took the swallows with apparently asymmetrical tail feathers a significantly longer time to find a mate than those with apparently symmetrical feathers. The swallows with asymmetrical feathers were also less likely to father a second clutch of eggs. This shows that female barn swallows take body symmetry into account when selecting a mate, a finding that has been echoed in similar studies of other monogamous bird species. In this case, body asymmetry proves to be a disadvantage to breeding males of species with this kind of directional mate selection.
In the case of the barn swallows, physical symmetry plays a key role in the reproductive success of the males, while within a lung, the asymmetric branching promotes an efficient use of the possible space. However, without the structural integrity symmetric development provides, the lung would not be able to operate at full capacity, and without the asymmetric lateralization of the brain, animals would be more vulnerable to predation. Though there are advantages and disadvantages to both symmetry and asymmetry, each has its place within the animal kingdom.